Hello students, today I am bringing for you a new chapter and that is molecular basis of inheritance. It is an important chapter of class 12th and in this chapter we are going to cover this topic and the topics are the DNA, the search for genetic material, the RNA world, the replication, transcription, genetic code, translation, regulation of gene expression, human genome project and DNA fingerprinting. So let's start this important chapter. First of all, this PDF. Molecular basis of inheritance. Here, as we are going through the molecular basis of inheritance, where the DNA and RNA, in the whole chapter we are going to cover only this topic. First of all, nucleic acids. We all familiar with it? Nucleic acids are the building block of genetic material, and DNA is the genetic material in most organisms. RNA is the genetic material in some viruses, because mainly RNA functions as messengers of the messages, or we can say data. First of all, structure of polynucleotide chain. Polynucleotides are the polymer of nucleotides, that are DNA and RNA, examples of polynucleotides. Here you can see poly means more than one, and here nucleotide 1, 2, 3 and 4. Combinedly they form polynucleotides, and DNA and RNA both are polynucleotides. And a nucleotide has three components, that are a nitrogenous base, as you can see here T for thymine, it is a nitrogenous base, a pentose sugar, ribose in RNA and deoxyribose in DNA, and a phosphate group here as you can see. Then nitrogenous bases are two types, purines and pyrimidines. Coming to purines, purines are two types, adenine, guanine, and pyrimidines are cytosine, thymine and uracil. Thymine 5-methyl-uracil present only in DNA and only uracil is present in RNA. Remember. Here, a nitrogenous base is linked to pentose sugar through a linkage that is called N-glycosidic linkage to form a nucleoside. Here you can see N-glycosidic bond is there where the nitrogenous base is combined with pentose sugar. Then nucleosides in RNA, that are adenosine, guanosine, cytidine, uridine, and nucleosides in DNA, that are deoxyadenosine, deoxyguanosine, deoxycytidine and deoxythymidine. Here the structures are there: adenosine, deoxyadenosine, guanosine, deoxyguanosine, cytidine, deoxycytidine and uridine or deoxythymidine. Next is again nitrogenous base combined with sugar and a phosphate group called nucleotide or deoxyribonucleotide. Like you can see, it is a nucleotide where nitrogenous base is present, pentose sugar is there and phosphate group is there. And in RNA, each nucleotide has an additional OH group at 2' position of the ribose. As you can see, it is a 2' position in this diagram where an additional OH group is present. Two nucleotides are linked through 3' to 5' phosphodiester bond to form dinucleotide. Here you can see phosphodiester bond. When more nucleotides are linked, it is formed polynucleotide. Obviously, it is a dinucleotide, and if more are combined, that is called polynucleotide. Then coming to the DNA. Before starting the DNA, let's see what your NCERT says here. Deoxyribonucleic acid DNA and ribonucleic acid RNA are the two types of nucleic acids found in living system. And DNA acts as genetic material in most of the organisms. RNA, though it also acts as genetic material in some viruses, mostly functions as a messenger of transformation. RNA has an additional role as well. It functions as adapter, structural and in some cases catalytic molecule. And in this chapter we are going to discuss about the structure of DNA, its replication, The process of making RNA from DNA, that is called transcription, the genetic code that determines the sequences of amino acids in proteins, and the process of protein synthesis, that is translation, and elementary basis of the regulations. The DNA. Here, what it is mentioned here, as you can see, DNA is a long polymer of deoxyribonucleotides. The length of DNA is usually defined as a number of nucleotides or a pair of nucleotides referred as a base pair present in it. This also is the characteristics of an organism. For example, a bacteriophage known as Phi174. This structure is called Phi, Phi, has 5386 nucleotides, and bacteriophage lambda has 48,502 base pairs. E. coli has 4.6 into 10 to the power 6 base pairs, and an haploid content of human DNA is 3.3 into 10 to the power 9 base pairs. Let's see the PDF what it says here. First of all, Friedrich Meister 1869 identified DNA and named it as nuclein. Remember, nuclein on 1869. James Watson and Francis Crick proposed the double helix model of DNA. As you can see, it is the double helical structure. It is the single one helix, it is the second helix. And who proposed it? James Watson and Francis Crick. It was based on X-ray diffraction data produced by Maurice Wilkin and Rosalind Franklin. Coming to structure, DNA is made up of two polynucleotide chains called in a right-handed fashion. As you can see, it is a polynucleotide chain 1 and it is the second one. Pitch of the helix, that is 3.4 nanometer or 334 angstrom, 3.4 nanometer. Number of base pairs in each turn is 10 and distance between adjacent base pair is 0.3 nanometer or 3.4 angstrom as it is mentioned here. Then backbone of DNA is formed of sugar and phosphate. The base project inside. The two chains have anti-parallel polarity, that is one chain has the polarity 5' to 3' and the other is 3' to 5', like this. 3' to 5', one is there, and 3' to 5', like this. Opposite one, that is called anti-parallel polarity. Then the base in two strands are paired through hydrogen bond forming base pairs. Like this, you can see it is a base pair A bond T, and here between the hydrogen bond is present here. Then A double bond T, that is two hydrogen bonds between the adenosine and thymine. And coming to cytidine and guanosine, there are three hydrogen bonds are there. Purines comes opposite to a pyrimidine. This generates uniform distance between the two strands. As you can see, purines comes always opposite to the pyrimidines, and this makes the uniform distance between the two strands. Remember, these two having three hydrogen bonds and these two having two hydrogen bonds between them. Here again the diagrams are there. As you can see, phosphate group, pentose sugar and nitrogenous base, and between this there are hydrogen bonds are present. Next, Irwin-Chargaff rule. What Irwin-Chargaff says? Now in DNA, the proportion of A is equal to T and the proportion of G always equal to C. Therefore, A plus G equal to T plus C, or we can say A plus G upon T plus C always equal to 1. Next, coming to the DNA, as you can see, Phi174, a bacteriophage, has 5386 nucleotides. Bacteriophage lambda has 48,502 base pairs. E. coli has 4.6 into 10 to the power 6 base pairs, and haploid content of human DNA is having 3.3 into 10 to the power 9 base pairs. Coming to calculation of length of DNA, length of DNA can be calculated by number of base pairs multiply distance between the two adjacent base pairs. Example, number of base pair in human is 6.6 10 to the power 9, and the length of DNA is 6.6 10 to the power 9 multiply 0.34 into 10 to the power minus 9 and approximately 2.2 meter. In E. coli, the length of DNA is 1.36 mm, or we can say 1.36 into 10 to the power minus 3 m. Therefore, the number of base pair is 4 into 10 to the power 6 base pairs. Next is packaging of DNA helix. Let's see what the NCERT says. Again, the same points are given. Structure of polynucleotide chain. There are three components. A nucleotide has three components: nitrogenous base, pentose sugar, ribose in case of RNA, and deoxyribose in DNA, and a phosphate group is there. There are two types of nitrogenous bases: purines and pyrimidines. Purines are adenine, guanine, and pyrimidines are cytosine, uracil, and thymine. Cytosine is common in both DNA and RNA, and thymine is present in DNA only. Uracil is present in RNA at the place of thymine. A nitrogenous base is linked to OH of 1' carbon pentose sugar through N-glycosidic linkage to form a nucleoside, such as adenosine or deoxyadenosine, guanosine or deoxyguanosine, cytidine or deoxycytidine, and uridine or deoxythymidine. When a phosphate group is linked to OH of 5' carbon of a nucleoside through phosphodiester linkage, a corresponding nucleotide is formed. Like so, here we can see a phosphate group is linked to OH of 5' carbon of a nucleoside through a phosphodiester linkage to form a nucleotide. Thus, a polymer has a 1' free phosphate moiety at 5' end of sugar, which is referred as the 5' end of polynucleotide chain. Similarly, the sugar has a free OH of 3' carbon group, which is referred as the 3' end of the polynucleotide chain. And the backbone is formed by sugar and phosphate. The nitrogenous base linked to sugar moiety project from the backbone. In RNA, every nucleotide residue has an additional OH group present at the 2' position of the ribose. Also in RNA, uracil is formed at the place of thymidine or 5-methyluracil. It is the another chemical name of thymine. DNA as acidic substance in nucleus was first identified by Friedrich Meister in 1869. He named it as nuclein. But at the time, because of technical limitations, so the structure of the proper DNA is remain elusive for a very long period of time. It was only in 1953 that James Watson and Francis Crick, based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin, proposed a very simple but famous double helix model of the structure for the structure of the DNA. And then comes Erwin Chargaff, and what it says that the ratio between adenine and thymine and guanine and cytosine are constant and always equals to 1. And coming to the double helix structure proposed by here Watson and Crick are follows, that are, it is made up of two polynucleotide chain where the backbone is constituted by sugar, phosphate, and the base project are inside. The two chains have an anti-parallel polarity, and it means if one chain has the polarity 5' to 3', then the other is 3' to 5'. The base in two strands are paired through hydrogen bonds, forming base pairs. Adenine forms two hydrogen bonds with thymine from opposite strand and vice versa. Similarly, guanine is bonded with cytosine with 3 hydrogen bonds. As a result, always purine comes opposite to pyrimidine, and this generates the approximately uniform distance between the two strands of the helix. Then the two chains are coiled in a right-handed fashion, and the pitch of the helix is 3.4 nanometer. A nanometer is the 1 billionth of a meter, that is 10 to the power minus 9 meter, and there are roughly 10 base pairs in each turn. Consequently, the distance between the base pair in a helix is approximately 0.34 nanometer, and the plane of one base pair stack over the other in double helix. This in addition to hydrogen bonds confers stability of the helical structure. And here a question is asked, why the distance between the two polynucleotide chains in DNA remains almost constant? And the answer is here, because the bases two strands of the DNA are paired through hydrogen bonds, forming base pairs. Adenine forms two hydrogen bonds with thymine from the opposite strand and vice versa. Similarly, guanine is bonded with cytosine with three hydrogen bonds. As a result of these bonds, the distance between the two polynucleotide chains in DNA remains almost constant. Then, Francis Crick also proposed the central dogma in molecular biology, which states that genetic information flows from DNA to RNA, then protein. From DNA to mRNA or messenger RNA, then in process of translation, it forms protein. This is called central dogma. In some viruses, the flow of information is in a reverse direction, that is from RNA to DNA, and the name is called reverse transcription. Remember. Packaging of DNA helix. Let's see what the PDF says. Packaging of DNA helix. Here. In prokaryotes, example E. coli, the DNA is not scattered through the cell. As you all know, DNA is negatively charged, so it is held with some positively charged proteins to form nucleoid. And see the diagram, nucleoid is there. And in eukaryotes, there is a set of positively charged basic proteins called histones. Positively charged basic proteins called histone. Histone are rich in positively charged basic amino acids residue that are lysine and arginine. 8 histones to form histone octamer. Here is the structure, histone octamer, and how in this circle how it is formed, as you can see. H2A, H2A, H2B, H2B, H3, H3 and H4, H4. Here H1 is there. Negatively charged DNA is wrapped around histone octamer to give nucleosome. So here the structure of nucleosome. The nucleosome molecule side view and is the top view. Then a typical nucleosome contains 200 base pairs. Therefore, total number of nucleosome in human is multiplied by 3.3 into 10 to the power 7. This is the total number of nucleosomes present in a human body. Here are the diagrams, as you can see. Then, nucleosomes constitute the repeating unit to form chromatin. Nucleosomes constitute the repeating unit to form a chromatin. Chromatin is a thread-like stained body. Nucleosome in chromatin is called beads on string. NCERT says beads on string. Here is a type of nucleosome. Here the second, third, fourth and fifth and furthermore. And they are attached like this and to form a chromatin. It is the structure of chromatin, as you can see. And NCERT says how it sees now, how it looks like. It looks like a beads on string, as you can see. Histone, nucleosomes are there, superly coiled, and if we make it free, then we can identify the nucleosomes and how it wrapped like an histone. Then in eukaryotes, chromatin is pegged, chromatin fibers formed, coiled and condensed at metaphase stage, and chromosomes are formed. Higher level packaging of chromatin requires non-histone chromosomal proteins. If the chromatin requires more or much complex structure, they require non-histone chromosomal proteins are present there. Here are the diagrams, you can see. Short region of DNA, double helix structure, 2 nanometer. Beads on string form the chromatin, 11 nanometer. 30 nanometer chromatin fiber packaged nucleosomes, 30 nanometer. Section of a chromosome in an extended form, 300 nanometer. Condensed section of chromosome, 700 nanometer. And finally, entire mitotic chromosome. Centromere is present, as you can see, it is 1400 nanometer. Then chromatins has two forms. Chromatins are two types, euchromatin or true chromatin or heterochromatin. Euchromatins are loosely packed, transcriptionally active regions of chromatin and its chains light, and heterochromatins are densely packed, inactive region of chromatin, its chains dark. As you can see, euchromatin is there, heterochromatin is there. It is a centromere. Clear? See the proper diagram. The search for genetic material. Experiments to prove which is the genetic material. Let's see what your NCERT says about this. Here, you can see taken the distance between the two consecutive base pairs as 0.3 nanometer. If the length of DNA double helix is a typical mammalian cell is calculated simply by multiplying the total number of base pairs with the distance between the two consecutive base pairs is 6.6 × 10⁹ base pair multiply 0.34 × 10⁻⁹ meter per base pair. It comes out approximately 2.2 meter. And then if the length of E. coli DNA is 1.36 mm, calculate the number of base pairs in E. coli. We already seen in the PDF, and it will be 4 into 10 multiplied 10 to the power 6 base pairs. Okay. Then in eukaryotes, the organization is much more complex comparing to prokaryotes. So for that, there is a set of positively charged basic proteins called histones are there. Histone proteins are there. A protein acquired charges depending upon the abundance of amino acid residue with charged side chain. Histones are rich in basic amino acid residue like lysine and arginine. Both amino acid residue carry positively charges in their side chain. Histones are organized to form a unit of 8 molecules called histone octamer, and the negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. A typical nucleosome contains 200 base pair of DNA helix. Nucleosome constitutes the repetitive unit of a structure in the nucleus called chromatin, and thread-like stained or colored bodies seen in nucleus. The nucleosome chromatin are seen as beads on string like this. Beads on string. It's the electronic microscope picture where the beads on string can be seen. Theoretically, how many such beads do you imagine are present in a mammalian cell? And it will be approximately 30 million nucleosomes are there. Then the packaging of chromatin at higher level requires additional set of proteins that collectively referred to as non-histone chromosomal proteins, NHC. In a typical nucleus, some regions of chromatins are loosely packed that are called euchromatin, and some are densely packed and stains dark that are called heterochromatin. Euchromatin is said to be transcriptionally active chromatin, whereas heterochromatin is inactive. Then the search of genetic material. Let's see the PDF. Here, experiments to prove which is the genetic material. So first of all, Griffith's transforming principle experiment, where the Frederick Griffith 1928 uses mice and Streptococcus pneumonia. Streptococcus pneumonia has two strains. One is the smooth strain or virulent, has polysaccharide mucus coat, causes pneumonia, and a rough strain, non-virulent, no mucus coat, do not causes pneumonia. As you can see, pneumonia, Streptococcus pneumonia are two types. One is the mucus coat is present, other is no mucus coat is there, and this does not causes pneumonia, but this causes pneumonia in mice. So Griffith taken and do some experiments to find out which one is the genetic material, because many scientists says that the protein is the genetic material, some says the RNA is the genetic material, and some says the DNA is the genetic material. So to find out which one is the genetic material, first experiment done by Frederick Griffith. And what it says, as you can see, first of all he taken live smooth strain which causes pneumonia and directly injected to the mice body, and the mice dies. As you can see, leaving smooth strain cells found in the heart muscles. Then living R strain are injected to the mice and the mouse is live. No bacterial cells found in the heart. Then what he do? He then heat killed the S strain. He provide the heat to the S strain, then injected to the mice and the mouse is alive and no bacterial cells found in the heart. Then he mixed the heat killed S strain with the live R strain and then mouse dies. The living S strain found in the cells of heart, and this is the experiments done by Griffiths. He concluded that some transforming principle, he given the name, some transforming principle transferred from heat killed S strain to the R strain. It enables the R strain to synthesize smooth polysaccharides coat and become virulent, because initially R strain is non-virulent, but when he added the heat killed S strain to the R strain enabled and become virulent. This is due to the transfer of genetic material. Then still this experiment does not conclude that which one is the genetic material. Then comes more scientists, biochemical characterization of transferring principle, where scientists like Oswald Avery, Colin MacLeod and MacLean McCarthy work to determine the biochemical nature of transferring principle in Griffiths experiment. They purified biochemicals, proteins, DNA and RNA from heat killed S strain using suitable enzymes. So these scientists, what they do, they use a protease, so that protease, it digests the protein content, RNA that digest RNA, DNA that digest DNA. So they purified the biochemicals, proteins, DNA and RNA from the heat killed S strain. Then they do the same experiment what Griffiths did, and what they find out, they discovered that the digestion protein and RNA using proteins and RNA did not affect transformation. So transforming substance was not a protein or RNA. Digestion of RNA with DNA inhibited the transformation. It means the DNA causes the transformation of R cell to the S cells. That DNA was the transforming principle, they concluded. Then again another experiment done by Hershey and Chase experiment, or we can say blender experiment. Hershey and Chase grew some bacteriophages viruses on a medium containing radioactive phosphorus P32 and some other medium containing radioactive sulfur S35. Bacteriophage means it is a type of bacteria which was infected by a virus, that is called bacteriophage. Then so they taken a medium containing radioactive phosphorus P32 and radioactive sulfur S35. Viruses grown in P32 got radioactive DNA because only DNA contains phosphorus. So the viruses grown in P32, they becomes radioactive DNA. Viruses grown in S35 got radioactive protein because proteins contain sulfur. Then what happened? These preparations were used separately to infect E. coli bacteria. After infection, the E. coli cells were gently agitated in a blender to remove the virus particles from the bacteria. Then the culture was centrifuged to separate the lighter virus particles from heavier bacterial cell. As you can see, medium containing bacteriophage protein is labeled S35, medium containing bacteriophage having DNA labeled P32. Then these are infected to the particular E. coli bacteria. These are the phages: bacterial cell, E. coli, radioactive protein, DNA. Then blending occur, and what the blending do? Blending remove the virus particles from the bacteria. So as you can see, protein coat viruses comes out from the bacteria. Then they centrifuge so that the lighter viruses particles from the heavier bacterial cells. Here you can see centrifugation occur, bacterial pellet, and here another pellet also here, radioactively phages of the DNA in pellet, and then Bacteria infected with viruses having radioactive DNA were radioactive, that is DNA has passed from viruses to the bacteria, and bacteria infected with the viruses having radioactive proteins were not radioactive, that is protein did not enter the bacteria from the viruses. So this proves that the DNA is the genetic material. And here as you can see, bacteriophage, radioactive S35 labeled protein capsule, radioactive P32 labeled as DNA. Both are the infected to the E. coli bacteria. It is the E. coli. It is also E. coli. And blending occur, then centrifugation occur. Here in the first one, what they found out? No radioactive S35 detected in the cell, plus radioactive S35 is a supernatant. And here radioactive P32 detected in the cell, no radioactivity in the supernatant, and this proves that the DNA is the genetic material, not the protein. Then a genetic material must have the following property, that are ability to generate its replication, chemical and structural stability, provide the mutation needs for evolution, and ability to express Mendelian character. And reason for stability of DNA, less reactive. Reason for mutability of RNA. DNA is the double-stranded, RNA is single-stranded. Presence of thymine, presence of uracil. Absence of 2' OH in sugar, presence of 2' OH in sugar. As you can see in ribose, in 2' end OH is present, but in deoxyribose only H is present. Then the RNA, thus the RNA is unstable. So RNA viruses, example QB bacteriophage, tobacco mosaic viruses, etc. mutate and evolve faster because RNA is unstable. That's why DNA strands are complementary. On heating, they separate. In appropriate condition, they comes together. In Griff's experiment, some property of DNA of the heat-killed bacteria did not destroy. It indicates the stability of DNA. As you can see in this diagram, it is a type of DNA. If we give heat, then you can see hydrogen bonds are breaking down and strands are unwinding. For giving more heat, both the strands are separated, and cooling, again double helix is reformed. Then for the storage of genetic information, DNA is better due to its stability, but for the transmission of genetic information, RNA is better. RNA can direct code for protein synthesis, hence can easily express the characters. DNA is dependent on RNA for protein synthesis. As you can see, DNA transcription may becomes RNA, translation proteins are formed, and from RNA reverse transcription occur, DNA is formed. Coming to RNA world. Before starting this, let's see the NCERT what it says. Here you can see the source for genetic material. Many scientists came, and even the discovery of nuclein by Friedrich Meister, the proposition of principal inheritance by Mendel were almost at the same time, but the DNA as genetic material took long to be discovered and proven by 1926. Mechanism for genetic inheritance had reached the molecular level, and previous discoveries by Gregor Mendel, Walter Sutton, Thomas Hunt Morgan, numerous other scientists had narrowed the search to the chromosomes located in the nucleus for the most cells, but the question of what molecule was actually genetic material had not been answered. Then you can see 1928 Friedrich Griffith came in a series of experiments with Streptococcus pneumonia, bacterium responsible for pneumonia, witnessed a miraculous transformation in the bacteria. And when the Streptococcus pneumonia bacteria are grown on a culture plate, some produce smooth shiny colonies or S, while the other produce rough colonies R. This is because S strain bacteria have a mucus coat, polysaccharide coat on them, while the R strain strain does not have any type of mucous coat. Mice infected with the S strain dies from pneumonia infection, but mice infected with the R strain do not develop pneumonia. Same experiment, Griffith was able to kill bacteria by hitting them, and he observed that the heat killed S strain bacteria injected in the mice did not kill them, and then when injected the mixture of heat killed S and live R bacteria, the mice died. He concluded that the R strain bacteria had somehow been transferred by the heat killed S strain. Some transforming principle, the transfer from the heat killed S strain, had enabled the R strain to synthesize a smooth polysaccharide coat and becomes virulent. Still, it is not properly clear that the genetic material. What is the genetic material? Then biochemical characterization. Here, Oswald Avery, Colin MacLeod and McLean McCarthy, 1933 to 44. They worked to determine the biochemical nature of transferring principle in the Griffith experiment. They purified biochemicals, all the proteins, DNA and RNA, from the heat-killed S cells to see which one could transfer live R cells into S cells. They discovered that DNA alone from S bacteria causes R bacteria to become transferred. They also discovered that the protein digesting enzymes, protease, RNA digesting enzymes, RNase, did not affect transformation. The transforming substance was not a protein or RNA. Digestion of with DNA that did the inhibit the transformation, suggesting that the DNA causes the transformation. And they concluded that the DNA is the hereditary material, but not all biologists were convinced. Then, finally, Alfred Hershey and Martha Cheese, 1952. They worked with viruses that infect bacteria that called bacteriophages. As I told you, viruses that infect bacteria called bacteriophages. The bacteriophage attached to the bacteria and its genetic material that enters the bacterial cell. The bacterial cell treated the viral genetic material as if its own and subsequently manufactured more viruses particles. Hershey Chase worked to discover whether it is protein or DNA from the viruses that enters the bacteria. They grew some viruses on a medium that containing radioactive phosphorus and some other on medium that contains radioactive sulfur. Viruses grown in the presence of radioactive phosphorus containing radioactive DNA but not radioactive proteins, because DNA contains phosphorus, not protein, but the proteins does not contain phosphorus. Similarly, viruses grown on radioactive sulfur contain radioactive proteins but not radioactive DNA, because DNA does not contain sulfur. Then radioactive phages were allowed to attach the E. coli bacteria. Then as the infection proceeded, the viral coat were removed from the bacteria by aggregating them in a blender. The virus particles were separated from the bacteria by spinning them in a centrifuge. Then bacteria which was infected with viruses that had radioactive DNA were radioactive, indicating that DNA was the material that passes from the virus to the bacteria, and bacteria that were infected with viruses that had radioactive proteins were not radioactive. This indicates that proteins did not enter the bacteria from the viruses, and DNA is therefore the genetic material that is passed from viruses to the bacteria. And here is the diagram. Come to the properties of genetic material, DNA versus RNA. Here, RNA is a genetic material, for example, tobacco mosaic virus, QB bacteriophages, etc. And coming to genetic material, it must fulfill the following criteria, that are: it should be able to generate its replica or do the replication, it should be stable chemically and structurally, it should provide the scope for slow changes, that is mutation, that are required for evolution, it should be able to express itself in the form of Mendelian characters. Then you can see, and from above all the experiments, RNA also known as catalytic, hence reactive. Therefore, DNA chemical is less reactive and structurally more stable compared to the RNA. Therefore, among the two nucleic acids, DNA is better genetic material comparing to RNA. Then, both DNA and RNA are able to mutate. In fact, RNA being unstable mutated at a faster rate. Consequently, viruses having RNA genome having shorter lifespan mutate and evolve faster. RNA can directly code for synthesis of proteins, hence can easily express the characters. DNA, however, is dependent on RNA for synthesis of proteins. The protein synthesis machinery has evolved around RNA. The above discussion indicates that both RNA and DNA can function as genetic material, but DNA, being more stable, is preferred for storage of the genetic information, and the transformation of the genetic information RNA is better. RNA world. Let's see what the PDF says about the RNA world. RNA was the first genetic material. It acts as genetic material and as well as catalyst. Essential life processes like metabolism, translation, splicing, etc. evolved around the RNA. DNA evolved from RNA for stability. Central dogma of molecular biology. Central dogma. It is proposed by Francis Crick. It states that the genetic information flows from DNA to RNA, then protein. And you can see from DNA to RNA and proteins by the process of transcription and translation. In some viruses, flow of information is in the reverse direction, that is from RNA to DNA. It is called reverse transcription. Then coming to the DNA replication. Replication is on the copy of DNA from parental DNA, obviously simply called replication. Watson and Crick proposed the semi-conservative model of replication. Watson and Crick, James Watson, Francis Crick. It suggests that the parental DNA strands act as template for the synthesis of new complementary strands, and after replication, each DNA molecule would have one parental and one new strand. Matthew Michelson and Franklin Stoll, 1958, proved the semi-conservative model. Here you can see in this diagram, the parental DNA, parental strands act as template, old parental strands and new strands are formed. The blue are the older one, red are the new one. It is the daughter DNA formed. Then Michelson and Stoll experiment. They prepared two culture media of E. coli. One preparation contains NH4Cl salt, where N15 is taken because it is the heavy isotope of nitrogen. So N15 was incorporated into both strands of the bacterial DNA and the DNA becomes heavier. Other preparation contains N14 salts. So N14 was incorporated in both strands and DNA becomes lighter. These two types of DNA can be separated by centrifugation in a CaCl density gradient, calcium chloride. As you can see, both the DNA are, you can see the density of N15 and the N14. Then they took E. coli cells from N15 medium and transferred to N14 medium. After one generation, that is after 20 minutes, they isolated and centrifuged the DNA. Its density was intermediate or hybrid, that is between N15 DNA and N14 DNA. As you can see, hybrid one between the N14 and N15. As I told you, N15 is heavy one and N14 is the lighter one. Then what happened? This shows that in newly formed DNA, one strand is older one or N15 type and the one strand is the new one N14 type. This confirms the semi-conservative replication. And after the second generation, that is after 40 minutes, there was equal amount of hybrid DNA and light DNA are formed, as I told here. Then Taylor and colleagues 1958 performed a similar experiment on Visia fava in plants, fava beans, using radioactive thiamidine to detect distribution of newly synthesized DNA in the chromosomes. It proved that the DNA in chromosomes also replicate semi-conservatively. Then DNA replication starts at the point called origin or ori. Here origin are present. O is mentioned here, as I told you. Here you can see in the diagram. Unit of replication with one origin is called replicon. It is a whole replicon. During replication, the two strands unwind and separate by breaking hydrogen bond in presence of an enzyme helicase. DNA helicase is present that breaks the hydrogen bonds and makes the stranded separate. Unwinding of DNA molecule at the point formed at Y shaped structure called replication fork. Then the separated strands are act as template for the synthesis of new strand. Here you can see presence of DNA helicase, it breaks down like this and Y shaped structure is present here like this, and these separated strands, they act as template strand. They are template or parental strand. The synthesis for the new strand. Then replicate in the 5 to 3' direction. Deoxyribonucleoside triphosphate, DATP, DGTP, DCTP and DTTP act as substrate and provide energy for polymerization. Here, firstly, a small RNA primer is synthesized in presence of an enzyme primase. In presence of an enzyme, DNA-dependent DNA polymerase, many nucleotides join with one another to primer strand to form a polynucleotide chain, that is new strand. As you can see, here old strands are present, new strands are present, and it is a leading strand, it is a lagging strand. And as you can see, first of all, an RNA primer is attached. Like you can see, presence of enzyme primase, it is RNA primer. RNA primer is present. Okay, and another enzyme DNA polymerase, many nucleotides join with one another to the primer strand and forms a polynucleotide chain or a new strand. Then during replication, one strand is formed as a continuous stretch 5' to 3' direction or continuous synthesis. This is called leading strand, here labeled leading strand, and the other strand is formed in a small stretches, that are Okazaki fragments in a 5' to 3' direction or discontinuous synthesis. Here you can see it is called lagging strand, the leading one. Directly it makes the strand, but here it becomes fragments. These fragments are called Okazaki fragments. Then the Okazaki fragments are then joined together to form a new strand by an enzyme DNA ligase. Then finally, after the formation of this strand, the lagging strand or fragments are attached to by DNA ligase, and the new strand is called lagging strand. If a wrong base is introduced in the new strand, DNA polymerase can do proofreading. Then you can see E. coli completes replication with 18 minutes, that are 2000 base pairs per second. In eukaryotes, the replication of DNA takes place at the S phase of the cell cycle, synthesis phase, DNA synthesis phase. Failure in cell division after DNA replication results in the polyploidy. Then come to the transcription. Before starting the transcription, let's see the same topic of the NCERT. Here RNA was the first genetic material, and how it works as a catalyst with reactive, unstable, and DNA has evolved from RNA with chemical modification that makes it more stable. Coming to replication, here a statement is given. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying of mechanism for genetic material proposed by Watson and Crick, 1953. And this scheme suggests that two strands would separate and act as template for the synthesis of the new complementary strands. And DNA molecule would have one parental and one newly synthesized strand. This scheme was termed as semi-conservative DNA replication. Experimental proof by taking E. coli. Matthew Michelson and Franklin Stahl performed the following experiment in 1958. They taken the E. coli in a medium containing NH4Cl, where N15 is taken, heavy isotope of nitrogen, and they also taken N14Cl, and by centrifugation calcium chloride is used, as you can see. Then the diagram here you can see N15, because of gravitational force, goes this side. It is heavy one, so it is present in the test tube in this portion, and after 20 minutes it becomes hybrid, and where you can see in generation 1 both the DNA are taken, and it comes to the middle portion, becomes hybrid, and after 40 minutes second generation N14, N15, N14 and N14 present, as you can see, light band, hybrid or dark band. Then E. coli divides in 20 minutes. Here a question is asked, if E. coli was allowed to grow for 80 minutes, then what would be the proportion of light and hybrid density of DNA molecule? Here the answer, after 80 minutes of the growth, the proportion of DNA molecule in E. coli would be as follows. First generation all strand will be hybrid, that is 50% N14 and 50% N15. Second generation 50% light density and 50% hybrid density of DNA molecules. Third generation 25% hybrid density, 75% light density DNA molecules. Fourth generation after 80 minutes 12.5% hybrid density and 87.5 light density DNA molecules are found. Then radioactive thymidine, another experiment involved the radioactive thymidine to detect distribution of newly synthesized DNA in the chromosome was performed on Vicia fava by Taylor and his colleagues in 1958, and this also proved the same one, that the DNA in chromosome also replicate semi-conservatively. Then the machinery and the enzymes. Here you can see, let's see, the main enzyme is referred to the DNA dependent DNA polymerase, since it is used as DNA template to catalyze the polymerization of deoxynucleotides. These enzymes are highly efficient enzymes as they have to catalyze polymerization on a large number of nucleotides in a very short time. E. coli, that is only 4.6 into 10 power 6 base pairs, compared to the human whose diploid content is 6.6 into 10 power 9 base pairs, complete the process of replication within 18 minutes. That means on an average rate of polymerization has to be approximately 2000 base pairs per second. Not only do these polymerases have to be fast, but they have to be catalyze the reaction with high degree accuracy. Any mistake during replication would result in the mutation. And furthermore, energetical replication is a very expensive process. Deoxyribonucleosides triphosphate serve as dual purposes. In addition to acting as substrate, they provide energy for polymerization reaction. The two terminal phosphates in a deoxynucleoside triphosphate and are highly energy phosphates, same as in case of ATP. In addition to the DNA dependent DNA polymerase, many additional enzymes are required to complete the process of replication with high degree of accuracy. And for a long DNA molecule, since the two strands of a DNA cannot be separated in its entire length due to very high energy requirements, the replication occurs with a small opening of the DNA helix referred to as replication fork. The DNA dependent DNA polymerase catalyzes polymerization only in one direction, that is 5 prime to 3 prime. Remember, only in one direction, 5 prime to 3 prime. This creates some additional complications at the replication fork. Consequently, one strand, the template polarity 3 prime to 5 prime, the replication is continuous, while the other one, which polarity 5 prime to 3 prime, discontinuous. So the discontinuously synthesized fragment are later joined by DNA ligases. Then the DNA polymerase on their own cannot initiate the process of replication. So for that, it requires a type of RNA. Also, replication does not initiate randomly at any place in DNA. Therefore, it is in a defined region in E. coli DNA where the replication originates, such as called origin of replication or simply called ORI. Then DNA, if needed to be propagated during recombinant DNA procedure, it requires a vector. The vector provides the origin of replication. And further, not every details of replication is understood well. In eukaryotes, the replication of the DNA takes place at the S phase of the cell cycle. The replication of DNA and the cell division cycle should be highly coordinated. A failure in cell division after DNA replication results into polyploidy. It is a chromosomal disability or anomaly. Transcription. Let's see in that PDF transcription here. This is the process of copying genetic information from one strand of DNA into RNA. That is called transcription. Here, as you can see in this diagram, it is DNA, template strand, coding strand. RNA polymerase is present here, and mRNA, it takes the copy of the information. Here, adenine from base pair with uracil instead of thymine. Next, the DNA dependent RNA polymerase catalyzes polymerization only in one direction, that is 5' to 3' direction, and 3' to 5' act as template strand and 5' to 3' act as coding strand. Here you can see coding strand, template strand, and both strands are not copied during transcription because the code for protein is different in both strands. This complicates the translation. If two RNA molecules are produced simultaneously, this should be complementary to each other. It forms a double-stranded RNA and prevents translation. That's why at a particular time, only a single strand codes for protein. Here, transcription occur like this, copy occur here. The coding strand and template strand remain. RNA is built for this. Transcription unit. It is a segment of DNA between the site of initiation and termination of transcription. It consists of three regions. So where the particular copy transcription started, it requires three regions where initiation occur, termination occur. First of all, a promoter, binding site of RNA polymerase. RNA polymerase has to be bind in the DNA, so a promoter is required located towards the 5' end, that is upstream. 5' end upstream promoter is added. Structural gene, region between promoter and terminator where the transcription takes place. Structural gene is present. A terminator, the site where the transcription stops, located towards the 3' end downstream. Terminator is present here. Then same points are given here. You can see promoter is present in this region, transcription initiation state, upstream of gene, RNA coding sequences, then terminator occur, and transcription termination site downstream of the gene. Then gene has a functional unit of inheritance. It is the DNA sequence coding for an RNA. It will be mRNA, RNA or tRNA. Cistron is a segment of DNA coding for polypeptide during protein synthesis. It is the largest element of a gene, cistron. Next, structural gene in a transcription unit has two types: monocistronic structural gene or split genes. It's in eukaryotes. Here coding sequence, exons or express sequence are interrupted by introns or intervening sequences. Exons appear in processed mRNA. Introns do not appear in processed mRNA. And another one is the polycistronic structural gene. It is seen in prokaryotes only. Here there are no split genes. As you can see, it is a type of gene. Eukaryotes found, it is exon introns are found. Exons are express sequence and introns are the intervening sequence. Exons appear in processed mRNA, introns do not appear. And coming to the prokaryote or polycistronic structural gene, it is in prokaryotes. Here there is no splitting genes, no exon, no intron. Remember. Then in bacteria, prokaryotes, synthesis of all type of RNA are catalyzed by a single RNA polymerase. Transcription occur in 3 steps: initiation, elongation, termination. Initiation occur, promoter is there, RNA polymerase and a sigma factor is present with the initiation site. Then elongation is occur, as you can see RNA is formed like this and the sigma factor is there, and termination RNA completely comes out, RNA polymerase and rho factor comes out, termination occur. First of all, initiation. Here the RNA polymerase enzyme binds at the promoter site of DNA. RNA polymerase enzyme binds at the promoter site of DNA. This causes the local unwinding of the DNA double helix. An initiation factor or sigma factor present in RNA polymerase initiate the RNA synthesis. Sigma factor is present. Initiation occur. Then elongation. RNA chain is synthesized in 5 to 3' direction. In this process, activated ribonucleoside triphosphate ATP, GTP, UTP or CTP are added. This is complementary to the base sequence in the DNA template. And it's the elongation process. Then termination. A termination factor or rho factor binds to the RNA polymerase, terminate the transcription. In bacteria, transcription and translation can be coupled. Translation begins before mRNA is fully transcribed because mRNA requires no processing to become active. Transcription and translation takes place in same compartment. No separation of cytosol and nucleus. That's why both are coupled. But in a eukaryotic cell, no coupling of transcription and translation. After transcription, translation occurs. Coming to the eukaryotes. In eukaryotes, there are two additional complexities. That are, there are 3 RNA polymerases and the primary transcript hnRNA contains exons and introns and are non-functional, and introns must be removed. Here the UT is there, untranslated region, primary transcript hnRNA, heteronuclear RNA. Here, 3 RNA polymerase in eukaryotes. First of all, RNA polymerase 1, it transcribes rRNA 28S, 18S and 5.8S. RNA polymerase 2, it transcribes heterogeneous nuclear RNA or hnRNA, it is the precursor of mRNA. And RNA polymerase 3, it transcribes tRNA, 5S rRNA and snRNA. SN means small nuclear RNA. As you can see, DNA, rRNA gene, RNA polymerase 1, RNA brings protein gene, RNA polymerase 2, mRNA brings it, tRNA, RNA polymerase 3, tRNA comes out. Then processing of primary transcript hnRNA. Here you can see, as I told you, introns do not need for this process, only exons are needed, so introns should be removed. That's why here what happened? Exon intron present here, RNA processing, spliced RNA for the exon 1 to 3 and the poly A tail fin present. Splicing from hnRNA, introns are removed by spliceosomes and exons are spliced together. But the introns are removed, remember. Spliceosomes splicing occur. Then capping. Capping means your nucleotide methyl guanosine triphosphate is added to the 5' end of the hnRNA. Remember, methyl guanosine triphosphate added as a cap. Then tailing or polyadenylation. Here the adenylate residue 200 to 300 are added at the 3' end. Tailing occur. Now it is the fully processed hnRNA called mRNA. Then genetic code. Before starting genetic code, let's see what your NCERT says. Here, transcription. The process of copying genetic information from one strand of the DNA into RNA is termed as transcription. Simply NCERT, this is the definition. So, here the same things are given. Coming to transcription unit, a transcription unit of DNA defined primarily by 3 regions: promoter, structural gene a terminator. Coming to the promoter 1, there is a convention in defining the two strands of the DNA in the structural gene of transcription unit. Since the two strands have opposite polarity, the DNA dependent RNA polymerase also catalyze the polymerization in only one direction, that is only 5 to 3 prime. Remember, the strands has the polarity 3 prime to 5 prime act as template strand and also referred as the template strand. The other strand, which is the polarity 5 to 3 prime, and the sequence of the same RNA except thymine at the place of uracil is displaced during the transcription, and this strand is referred as coding strand. All the references point while defining a transcription unit is made with the coding strand. To explain the point, a hypothetical sequence from a transcription unit is represented below. As you can see, 3 prime, 5 prime template strand, 5 prime to 3 prime coding strand, and these are the hydrogen bonds between them 2 and 3, and they are oppositely present. Here, schematic diagram: transcription unit, promoter is there, transcription start, structural gene made, template strand, coding strand, terminator is present, and the RNA comes out like this. The promoter and terminator flank the structural gene in a transcription unit. The promoter is said to be located towards the 5 prime end, or we can say upstream, and it is a DNA sequence that provides binding site of the RNA polymerase. Binding site of RNA polymerase, and it is the presence of a promoter in a transcription unit that also defines the template and coding strand. By switching its position with terminator, the definition of coding and template strand could be reversed. The termination is located towards the 3 prime end downstream of the coding strand. Then transcription unit and the gene. A gene is defined as a functional unit of inheritance. We already know, though there is no ambiguity that the genes are located in the DNA, it is difficult to literally define a gene in terms of DNA sequence. The DNA sequence coding for tRNA or rRNA molecule also define a gene. However, by defining a cistron as a segment of DNA coding for polypeptide, a structural gene in transcription unit could be said as monocistronic, mostly in eukaryotes, or polycistronic, mostly in bacteria or prokaryotes. In eukaryotes, the monocistronic structural gene has interrupted coding sequences. The genes in eukaryotes are split. The coding sequences or expression sequence are called exons, and exons are said to be those sequences that appears in mature or processed RNA. The exons are interrupted by introns. Introns or intervening sequences do not appear in mature or processed RNA. The split gene arrangement further complicates the definition of a gene in terms of DNA segment. And then inheritance of a character is also affected by promoter and regulatory sequences of a structural gene. Hence, sometimes the regulatory sequences are loosely defining as regulatory genes, even though these sequences do not code for any RNA or protein. And then coming to types of RNA and the process of transcription. In bacteria, there are 3 major types of RNA: mRNA or messenger RNA, tRNA or transfer RNA, rRNA or ribosomal RNA. All 3 RNA are needed to synthesize a protein in a cell. The mRNA provides the template, tRNA brings amino acids and reads the genetic code, and rRNA play structural and catalytic role during translation. There is a single DNA dependent RNA polymerase. That catalyzes transcription of all type of RNA in bacteria. RNA polymerase binds to promoter and initiates transcription, or we say initiation. It uses nucleoside triphosphate as a substrate. And polymerases in a template depending fashion following the rule complementarity. It somehow also facilitates the opening of the helix and continuous elongation. Only a short stretch of RNA remains bound to the enzyme. Only the polymerase reaches the terminal region, the nascent RNA falls off, so also the RNA polymerase. This results the termination of transcription. And an intriguing question is that how the RNA polymerase is able to catalyze all the three steps, which are initiation, elongation and termination. And the answer is, the RNA polymerase is only capable of catalyzing the process of elongation. It associates transiently with initiation factor and a termination factor, sigma factor and a rho factor, to initiate and terminate the transcription. Association with this factor alter the specificity of the RNA polymerase to either initiate or terminate. Simple. In bacteria, since the mRNA does not require any processing to become active, and also since transcription and translation takes place in the same compartment, many times the translation can begin much before the mRNA is fully transcribed. Consequently, the transcription and translation can be coupled in bacteria. But in eukaryotes, there are two additional complexities. There are at least 3 RNA polymerases in the nucleus in addition to the RNA polymerase found in the organelles. There is a clear-cut division of labor. RNA polymerase 1 transcribes rRNAs that are 28S RNA, 18S and 55.8S, where the RNA polymerase 3 is responsible for transcription of tRNA, 5srRNA and snRNA, small nuclear RNA. The RNA polymerase 2 transcribes precursor of mRNA, the heterogeneous nuclear RNA or hnRNA. Then the second complexity is that the primary transcripts contains both the exons and introns, which are non-functional. Hence, it is subjected to the process called splicing, where the introns are removed and exons are jointed in a defining order. hnRNA undergoes additional processing called as capping and tailing. In capping, an unusual nucleotide, methyl guanosine triphosphate, is added to the 5' end of the hnRNA, and in tailing, adenylate residue 200 to 300 are added at the 3' end in the template independent manner. It is the fully processed hnRNA, now called the mRNA, that is transported out of the nucleus for translation. The significance of such complexities is now beginning to be understood. The split gene arrangements represent the probability of an ancient feature of the genome. This presence of intron in reminiscent of antiquity and the process of splicing represent the dominance of RNA world. Then the genetic code. Let's see about the PDF and in genetic code here. Genetic code means it is a sequence of nucleotides or nitrogenous bases in mRNA that contains information for protein synthesis or translation. The sequences of 3 bases determining a single amino acid is called codon. This codon is present, as you can see. Codes are there. George Gamio suggested that for coding 20 amino acids, the code should be made up of 3 nucleotides. Thus, there are 64 codons. To code 20 amino acids, the code should be made up of 3 nucleotides. So, there are 64 codons are there. Here you can see from this diagram. Then Horgobin Khurana developed the chemical method in synthesizing RNA molecules with defining combination of the bases, homopolymer or copolymers. Then Marshall Nirenberg developed cell-free system for protein synthesis, and Severo-Ochoa enzyme, polynucleotide phosphorylase, is used to polymerize RNA with defining sequences in a template independent manner. 20 types of amino acids involved in translation. Here are the given: alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, tyrosine and valine. And in shortcut, names are also mentioned. Here the genetic code, codons for various amino acids, the genetic code dictionary, as you can see. That is an important one. Then codons is triplet or 3-letter code. 61 codons code for amino acids. 3 codons UAA, UAG and UGA do not code for any amino acids. They are act as stop codons or termination codon or nonsense codons. AUG has a dual function. It codes for methionine and acts as a initiator codon. In eukaryotes, methionine is the first amino acid and formal methionine in prokaryotes. An amino acid is coded by many codons except AUG for methionine and UGG for tryptophan. Such codons are called degenerate codons. So as I told you, AUG is the initiator codon, and as you see, triple codes are given. AUG present, initiator codon, and UA is a stop codon. Three stop codons are there: UAA, UAG, and UGA. Then, no punctuation between adjacent codons, comma less code. The codons is read in mRNA in a contiguous fashion. Genetic code is universal. Example from bacteria to human. UUU codes for phenylalanine. Some exceptions are found in mitochondrial codons and in some protozoans. Genetic coding is non-overlapping. Genetic code is unambiguous and specific. That is, one codon specific only one amino acids. As you can see, no comma between the adjacent codons and no here overlapping is also there, but you can see there are two codons and they are present in the contiguous fashion. Then, mutation genetic code. Relationship between genes and DNA are best understood by mutation studies. Deletion and rearrangement in a DNA may causes loss or gain of a gene and so a function. Insertion or deletion of one or two bases changes the reading frame from the point of insertion or deletion. It is called frame shift insertion or deletion mutation. Here you can see it is a normal sequence. Insertion of one base, G is added, reading frame changed, obviously one is added, then reading frame is changed, then normal deletion of one base and this is deleted and the reading frame is changed, as you can see. Then insertion and deletion of three or its multiple bases inserted or deleted one or multiple codon, the reading frame remains unaltered from the point onwards. Hence, one or multiple amino acids are inserted or deleted. It proves that codon is a triplet and can read contiguously. Here you can see directly three bases are added. GAG are added. Reading frame unaltered. Normal sequence. Deletion of this base is deleted, as you can see. Again, reading frame is unaltered. If one is added, then it is changed. But it is one is added or deleted, that three are added or deleted, then reading frame is unchanged. Then types of RNA, mRNA or messenger RNA, it provides template for translation, protein synthesis. rRNA or ribosomal RNA, the structural and catalytic role during translation. Example 23, srRNA. In bacteria act as ribozyme. tRNA or transfer RNA or sRNA or soluble RNA brings amino acids for protein synthesis and reads the genetic code. See the diagram: mRNA, rRNA, tRNA. Then Francis Crick postulated presence of an adapter molecule that can read the code and to the link with the amino acids. tRNA is called adapter molecule because it has an anticodon loop that has bases complementary to the codon, an amino acid acceptor end to which amino acids binds, ribosome binding loop, enzyme binding loop. Here you can see it is a type of tRNA where amino acid acceptor end, enzyme binding loop, ribosome binding loop, and an anticodon loop. It is the cloverleaf model of tRNA. Then for initiation, there is another tRNA called initiator tRNA. There are no tRNA for stop codon. Secondary structure of tRNA looks like cloverleaf and 3D structure looks like inverted L. Here you can see 3D model, inverted L like shape, and it is a cloverleaf structure like this. Okay, cloverleaf model. Then for initiation there is another tRNA called initiation RNA. Okay, same points are given, structures are different, you can see diagrams are changed. Then translation or protein synthesis. Let's see the PDF what it says. Sorry, NCERT. Genetic code, it is George Gamma, a physicist, who argued that since there are only 4 bases and if they have to code for 20 amino acids, the code should be constituted by a combination of bases. Hence, in order to code for all the 20 amino acids, the code should be made up of 3 nucleotides. This was a very old proposition, and then providing proof that the codon was triplet, scientist came Hargobind Khurana. Instrumental in synthesizing RNA molecule with defining combination of bases, homopolymer and copolymer. Then Marshall Nirenberg came. Selfie system for protein synthesis finally helped the code to be deciphered, and severoenzyme polynucleotide phosphorylase was also helpful in polymerizing RNA with defining sequence in a template independent manner. Then the particular checkerboard is present, like this you can see. The salient feature of genetic code are as follows, and what are they? Codon is triplet. 61 codons code for amino acids. 3 codons do not code for any amino acids, hence they are called stop codons. Some amino acids are coded by more than 1 codon, hence the code is degenerate. The codon is read in an mRNA in a contiguous fashion. There are no punctuations. The code is nearly universal. For example, from bacteria to human, you would code for phenylalanine. Some exceptions to the rule have been found in mitochondrial codon and in some protozoans. AUG has dual function. It codes for methionine. It also acts as the initiator codon. And UAA, UAG, UGA are the stop or terminator codons. Then mutation and genetic code. Here you can see mutation occur. When the mutation occur, here the relationship between the genes and DNA are best understood by mutation studied. Effect of large deletion or rearrangement in a segment of DNA are easy to comprehend. It may result in loss or gain of a gene, so as a function. This effect of a point mutation will be explained here. Point mutation is the change of single base pair in the gene for beta globin chain that results in the change of amino acid residue glutamate to valine. It results in the disease condition of called sickle cell anemia. Effect of point mutation that insert or deletes a base in a structural gene can be understood by the following example. You can see single letter is given, single code entered here like this, and the whole line is altered, as you can see. Then the whole 3 codons are inserted, and the reading steps are same. Like this, here is inserted, the 3 codon BIG is inserted, and here this one is the deleted. Okay, here the deleted. So such deletion methods are called frame-shift insertion or deletion mutation. Insertion or deletion of a 3 or its multiple bases inserted or deleted in one or multiple codon, hence one or multiple amino acids, and reading frame remains unaltered form that points towards tRNA or the adapter molecule. Francis Crick that there has to be mechanism to read the code and also to link it to the amino acids, because amino acids have no structural specialties to read the code uniquely. The tRNA, then called sRNA, known before the genetic code was postulated. tRNA has an anticodon loop that is bases complementary to the codes, and it also has an amino acid acceptor end to which it binds the amino acids. tRNA are specific for each amino acids. For initiation, there are another specific tRNA referred initiator RNA, initiator tRNA. There are no tRNA for stop codons, remember, and it looks like clover leaf in 2D structure. and in 3D structure it looks like inverted L. Then translation. See the PDF what translation says. Translation or protein synthesis. It is a process of polymerization of amino acids to form polypeptide based on the sequence of codons in mRNA. It takes place in ribosomes. Ribosomes consist of structural RNA and about 80 type of proteins. Ribosomes also act as a catalyst. 23S rRNA in bacteria and some enzymes is the enzyme ribozyme for the formation of peptide bond. Peptidyl transferase enzyme in large subunit of ribosomes. Coming to the steps of translation, here first step is the charging of tRNA, initiation, elongation, termination. First of all, charging of tRNA or amino acylation. Here what happened? Formation of peptide bond needs energy obtaining from ATP. From this, amino acids are activated, amino acid plus ATP, and linked to their cognate tRNA in presence of amino acyl tRNA synthesis. Thus, tRNA becomes charged. Here you can see amino acid acceptor site. It is the anticodon loop, anticodon. Amino acyl tRNA synthesis occur where the ATP is added and amino acetyl tRNA or alanine tRNA ester bond is formed and it is called the tRNA is charged. Then initiation. In the small subunit of ribosome binds to mRNA at the start codon AUG. Here AUG is the start codon. Now large subunit binds to small subunit to form initiation complex. Initiation complex is formed. Large subunit consists of amino acid tRNA binding site A site and peptidyl site P site. A site and P site are there. The initiator tRNA which carries methionine binds on P site. Methionine binds on P site. It anticodon UAC recognizes start codon AUG. Anticodon UAC start codon AUG. Initiator tRNA and methionine is attached. You can see in the diagram. Then elongation. Second amino acyl tRNA binds to the A site of ribosome. Its anticodon binds to the second codon on the mRNA and a peptide bond is formed between first and the second amino acids in presence of peptidyl transferase. Here you can see peptidyl transferase, peptide bond is formed and first amino acid and its tRNA are broken. First amino acid and its tRNA are broken. This tRNA removed from P site and second tRNA from A site is pulled to P site along with the mRNA. This is called translocation. This process are repeated for other codons in mRNA. During translation, ribosomes move from codons to codon. As you can see, ribosomes move from one codon to another codon like this. It started elongated. Then termination. When a releasing factor binds to the stop codon, here you can see stop codon is there. UAA is a stop codon. The releasing factor binds like this. Translation terminates. The polypeptide and tRNA released from the ribosome. Polypeptide and tRNA released from the ribosome. The ribosome dissociate into large and small subunit. It dissociate like this, as you can see, and the polypeptide chain released. Then a group of ribosomes associated with a single mRNA for translation is called polyribosome or polysomes. Here you can see the diagram. An mRNA has additional sequence that are not translated or untranslated regions or UTR. UTR are present at both 5' or before start codon. As you can see, UTR are there before start codon and 3' after stop codon. Stop codon and after it is present. They are required for efficient translation process. Then, see the diagram. Larger subunit, ribosome, smaller subunit, tRNAs and your peptide transferase mRNA is formed. Initiation, it occur like AUG. Then initiation, methionine tRNA binds at the P site. Methionine tRNA binds at the P site. Then elongation, second tRNA binds at the A site, that is thyroxine binds at the A site. Peptide bond is formed. This bond, peptide bond is formed. Then elongation, translocation from A to P site. Again translocation for A to P site. Another is binded here. As you can see, tRNA is binded here. And peptide transfer is also here. Then in elongation, as you can see, this is formed all one and it started elongated. Then termination occur, stop codon is there, releasing factor binds at the stop codon UAA and it breaks down. As you can see, larger subunit and smaller subunit of ribosome breaks down and this is the required RNA is formed. Regulation of gene expression. Before starting this, let's see your NCERT what it says. Same points are given what we see in that PDF. Translation is the process of polymerization of amino acids from a polypeptide. Then the order and sequence of amino acids are defined by the sequence of bases in the mRNA. The amino acids are joined by bond which is known as peptide bond. Formation of peptide bond requires energy. Therefore, first phase itself amino acids are activated, appearance of ATP and linked to the cognate tRNA and called charging of tRNA or amino acylation of tRNA. Then the same process given, you can see the ribosomes consist of structural RNA and about 80 different proteins. In its inactive state, it exists only two subunits, a larger subunit and a small subunit. When the small subunit encounters an mRNA, the process of translation of the mRNA to protein begins. There are two sites in the large subunit for subsequent amino acids to bind and thus to be close enough to each other for the formation of peptide bond. The ribosome also acts as catalyst. 23S rRNA in bacteria is an enzyme. Ribosome is there for the formation of peptide bond. Then a translation unit in mRNA is the sequence of RNA that is flanked by start codon AUG and stop codon codes for polypeptide. mRNA also has some additional sequences that are not translated and referred as untranslated regions UTR. The UTR are present at the both 5' end before start codon and 3' end after stop codon. They are required for efficient translation process. Then the process given: initiation, then formation, elongation and finally termination. At the end, a releasing factor binds to the stop codon, terminate translation, releasing the complete polypeptide from the ribosome. Then is regulation of gene expression. Let's see that PDF here. Here you can see in eukaryotes gene expression occurs by following level. First of all, transcriptional level, that is formation of primary transcript, processing level, splicing or capping, transport of mRNA from nucleus to the cytoplasm, and fourth is translation level, formation of a polypeptide. Here you can see genes on DNA, snRNA, introns, exons are there, mRNA only exons are there, and then proteins are formed. And these are the steps you can see. Then The metabolic, physiological and environmental conditions regulate the gene expression. Examples: in E. coli, the beta-galactosidase enzyme hydrolyzes lactose into galactose and glucose. In the absence of lactose, the synthesis of beta-glycosidase stops. The development and differentiation of embryo into an adult are a result of the expression of several set of genes. If a substrate is added to the growth medium of a bacteria, a set of genes is switched on to metabolize it. It is called induction. When a metabolite or product is added, the genes to produce it are turned off. This is called repression. Then operon concept. Each metabolic reaction is controlled by a set of genes. All the genes regulating a metabolic reaction constitute an operon. Example: lac operon, trip operon, ara operon, his operon and val operon, etc. Here you can see, but we will go through lac operon only. Lac operon is the operon controlling lactose metabolism. Lactose metabolism, simply called lac operon. It is proposed by Francois Jacob and Jacques Monod. Lac operon consists of a regulatory or inhibitor gene, like this, code for repressor protein, 3 structural gene, Z gene, Y gene and A gene, ZYA called structural genes, and an operator region O. Then genes in the operon functions together in the same or related metabolic pathway. Role of the structural genes. The Z gene codes for beta-galactosidase. Z gene codes for beta-galactosidase. It hydrolyzes lactose to galactose and glucose. Y gene, it codes for permease. It increases permeability of the cell of the beta-galactosidase. A gene codes for transacetylase. Lac operon. In the absence of inducer, if there is no lactose, lac operon remains switch off. Obviously, the regulator gene synthesizes mRNA to produce the repressor protein. This protein binds to the operator region and block RNA polymerase movement. So the structure genes are not expressed and lac operon is switched off. Then if the inducer is present, means lactose is present, like this, inducer is there. If lactose provided in the growth medium, the lactose is transported in E. coli cells by the action of permease. Lactose binds with repressor protein, so repressor proteins cannot bind to the operator gene. The operator region becomes free and induces the RNA polymerase to bind to the promoter gene. Then the transcription starts. Then glucose or galactose cannot act as inducer for lac operon. Only lactose is used as the inducer. Regulation of lac operon by repressor is called negative regulation. Regulation of lac operon by repressor is called negative regulation. Lac operon is under control of the positive regulation as well. Here you can see, inducer is there, lactose is present. Then inducer, what it do? Repressor mRNA is formed, proteins are formed. Then it directly goes to the operator region and then transcription occur, where the Z produce beta-galactosidase, Y produce permease, A produce transacetylase and lactose metabolized. If the switch on cases. In the Human Genome Project, let's see your NCERT what it says about this. Here, regulation of gene expression refer to a very broad term that may occur at various levels, and in eukaryotes regulation could be exerted at transcriptional level, formation of primary transcript, processing level, regulation of splicing, transport of mRNA from nucleus to the cytoplasm, and translational level. See, the genes in a cell are expressed to perform a particular function or a set of functions. For example, if an enzyme called beta-galactosidase synthesized by E. coli is used to catalyze hydrolysis of disaccharides, lactose into galactose and glucose, the bacteria use them as a source of energy. Simple. Hence, if the bacteria do not have lactose around them to be utilized for energy source, they would no longer require synthesis of enzyme beta-galactoside is simple. Therefore, in simple term, the metabolic, physiological and environmental conditions that regulate the expression of genes. In prokaryotes, control of the rate of transcriptional initiation is the predominant site of the control of gene expression. In a transcriptional unit, the activity of RNA polymerase at a given promoter is in turn regulated by interaction with accessory proteins, which affect the ability of recognizing start site. These regulatory proteins can act both positively and negatively. The accessibility promoter region, prokaryotic DNA, is in many cases regulated by the interaction of proteins with sequence termed as operator. So, with this, for lactose, lac operon says it interacts specifically with the lac repressor only. Then, lac operon. Francois Jacob, a biochemist, and Jacob Monod, two scientists are there. They were the first to elucidate a transcriptionally regulated system. In lac operon, a polycystonic structural gene is regulated by common promoter and regulatory genes, and such arrangement is called operon. There are other examples: lac operon, tryptophan operon, R operon, his operon, and val operons, etc. Lac operon consists of one regulatory gene, that is I gene. Here I does not mean inducer, rather it is called inhibitor, and three structural genes Z, Y, and A. I codes for the repressor of the lac operon, Z gene codes for beta-galactosidase, and which is primarily responsible for the breaking down of the disaccharides lactose into galactose and glucose. Y gene codes for permease, which increase the permeability of the cells to the beta-galactosides, and A gene codes for transacetylide. Then see the diagram. In the absence of repressor, inducer means lactose is not in the outside environment of the E. coli bacteria. Then what happened? I gene makes repressor mRNA, and repressor is formed, and repressor is the operator. Repressor binds to the operator region and prevent the RNA polymerase to transcribe the operand. So there is no use of this. Then if the inducer is present, means lactose is present, then what happen? Repressor mRNA, though it makes the repressor, inducer it makes the inactive the repressor. So it does not bind with the operator, and the process start, and the functional enzymes, as you can see, they form the particular beta-galactoside is permease, translated through translation. Then lactose is the substrate for the enzyme beta-galactosidase, and it regulates switching on and off the operon, hence it is termed as inducer. Remember, glucose and galactose cannot act as inducer for lac operon. Regulation of lac operon by repressor is referred to as negative regulation. Human Genome Project. Let's see your PDF. Here you can see the entire DNA is the haploid set of chromosomes of an organism is called a genome. In human genome, DNA is packed in 23 chromosomes. Human genome contains about 3 into 10 to the power 9 base pairs. Human Genome Project 1990s to 2003 was the first mega project for the sequencing of the nucleotides and mapping of all the genes in human genome. Human Genome Project was coordinated by US Department of Energy and the National Institute of the Health. Goals of Human Genome Project. First goal is to identify all the estimated genes in the human DNA. Second, sequencing of 3 billion chemical base pairs of human DNA. Third, store the formation in database. Fourth, improve tools for data analysis. Fifth, transfer of related technologies to other sector. And the last one is the address the ethical, legal and social issue ELSI that may arise from the project. Here you can see genome, chromosomes are the DNA and genes are formed. Then methodologies. It involves two major approaches. First of all, expressional sequence tag EST, where focused on the identifying all the genes that are expressed as RNA. Like they only see the exons like this. Only read this. EST, expressed sequence tag. Then sequence annotation, that are sequencing whole set of genome containing all the coding and non-coding sequences that are assigning different region in the sequence with function. Means introns are also present. Next procedure. First, isolate DNA from a cell, convert into random fragments, means cuts. As you can see, DNA is isolated, then cuts down like this. Clone in a host bacteria or yeast using vector BAC and YAC for amplification. Sequencing of fragments using automated DNA sequencer using Friedrich Sanger method. Arrange the sequence based on overlapping regions. Alignment of sequences using computer programs. BAC stands for bacterial artificial chromosomes. YAC stands for yeast artificial chromosomes. Full form is asked in the paper. Here you can see procedure. DNA extracted from liver cell, prepared library. As you can see, hundreds of millions of genomic DNA fragments on a surface. Then single genome fragment makes thousands of copies. Genomic sample into sequencing machine, then computerized analysis of data. Then Sanger has also developed a method for sequencing of amino acid in proteins. DNA is converted to random fragments as there are technical limitations in sequencing very long piece of DNA. Human Genome Project was closely associated to the bioinformatics. Bioinformatics means application of computer science and information technology to the field of biology and medicine, called bioinformatics. Then, of the 24 chromosomes, 22 autosomes and XY, the last sequenced one chromosome 1 May 2006. Genetic and physical map on the genome were generated using information on polymorphism of restriction endonuclease recognition site and some repetitive DNA sequences, microsatellites. DNA sequence also have been done in bacteria, yeast. Next, Senora habitus elegans, a free living non-pathogenic nematode, Drosophila, plants, rice and Arabidopsis, etc. Silent features. Human genome contains 3164.7 million nucleotide bases. Total number of genes is about 30,000. Every gene consists of 3000 base pairs, but size vary. Largest known human gene is dystrophin on X chromosome, contains 2.4 million base pairs. Next, 99.9% nucleotide bases are same in all people. Only 0.1%, that is 3 into 10 power 6 base pair different, makes every individual unique one. Coming to functions, of over 50% of discovered genes are unknown. Chromosome 1 has most gene, that is 2968, and Y has the fewest, that is 231. Less than 2% of the genome codes for protein. Next, very large portion of human genome is made up of repeated sequences. These are stretches of DNA sequences that are repeated many times. They have no direct coding function. They shed light on chromosome structure, dynamics and evolution. About 1.4 million locations have single base DNA differences. They are called SNP, single nucleotide polymorphism, or SNPs. This helps to find chromosomal locations for disease associated sequences and tracing human history. DNA fingerprinting. Before starting this, let's see what your NCERT says. Human Genome Project. Same points are given. Launched in the year 1990. It is a mega project and approximately 3 into 10 to the power 9 base pairs. And if the cost of sequencing require $3 per base pair, then the estimated cost will be $9 billion US. And during storage, if each page of the book contains 1000 letters and each book contain 100,000 pages, then 3300 such books would be required to store information of DNA sequences from a single human cell. And human genome project was closely associated with the rapid development and new area in biology called bioinformatics. Goals we already know: identify all the approximately 20,000 to 25,000 genes in human DNA, determine the sequence of the 3 billion chemical base pairs that make up the human DNA, store this information in database, improve tools for data analysis, transfer related technologies to other sectors such as industries, address the ethical, legal and social issue that may arise from the project. It was a 13-year project coordinated by US Department of Energy and the National Institute of the Health. During early years of the HGP, the Wellcome Trust became a major partner. Additional contribution came from Japan, France, Germany, China and others. The project was completed in 2003, and knowledge of the DNA variation among individuals can lead to revolutionary new ways to diagnose, treat and someday prevent the thousands of disorders that affect human beings. Many non-human models organisms such as bacteria, yeast, Caenorhabditis elegans, Drosophila plant have also been sequenced. Methodologies to approaches are there: EST and a blind approach. Here sequence annotation is there, BAC and YAC are also here, full form is given, and Friedrich Sanger, it is also mentioned about him. He is also credited for developing methods for determination of amino acid sequence in protein. Then the sequence of chromosome 1 was completed only in May 2006. It is the last of the 24 human chromosomes, 22 autosomes and XY to be sequenced. Another challenging task was assigning the genetic and physical map on the genome. This was generated using information on polymorphism of restriction endonuclease recognition site and some repetitive DNA sequences known as microsatellites. We'll go through about the restriction endonuclease recognition site in further chapter afterwards. The silent features, you can see all the points that we all discussed in that PDF. Applications and future challenges. DNA fingerprinting. I think it's the last topic. Let's see the PDF, what it says. DNA fingerprinting or DNA profiling. The technique to identify the similarities and differences of the DNA fragments of two individuals, developed by Alex Jeffrey, 1985. DNA carries some non-coding repetitive sequences. We all know. Number of repeats is specific from person to person. Example, you can see person 1 has this repeats, person 2 this type of repeat, person 3 9 repeats and 10 repeats, as you can see. Then repetitive DNA can be separated from bulk genomic DNA as different peak during density gradient centrifugation. You can see the uniform one, and during after centrifugation gradients are different, satellite, bands, major peak. The bulk DNA forms a major peak and the small peaks are called satellite DNA. Then satellite DNA is classified as microsatellite, mini satellites, etc. based on base composition, AT reach and GC reach, length of segments and numbers of repetitive units. A DNA sequence which is tandemly repeated in many copy numbers are called variable number tandem repeats, VNTR. VNTR belongs to many satellite DNA. As you can see, STR short random repeat, STR where repeated 8 times, repeated 20 times, and many satellite VNTR, size of the each repeat hundreds of base pairs, repeated 4 times. Then in a person, copy number varies in each chromosome. Two alleles, paternal and maternal, of a chromosome contains different copy number of the VNTR. As you can see, two alleles of a chromosome, paternal and maternal. VNTR is specific from person to person. The size of VNTR varies from 0.1 to 20 kilobase. Then, any difference in the nucleotide sequence observed in a population is called DNA polymorphism. Polymorphism is higher in non-coding DNA sequence because of mutation in these sequences may not affect an individual's reproductive ability. These mutations accumulate generation to generation, causing polymorphism. Polymorphism has great role in evolution and speciation. Then, Southern blotting technique. Isolation of DNA from any cell or blood stain, semen stain, saliva, hair root, bones, skins, etc. DNA sample can be collected and isolated. Then digestion of DNA by restriction endonuclease. The DNA can be digested. Then third is the separation of DNA fragment by gel electrophoresis. Then treat the DNA fragments with alkali solution NaOH to denature DNA bonds in the gel into single strands. Then fourth is transferring or blotting of DNA fragments to synthetic membranes such as nitrocellulose or nylon. Then hybridization using radioactive labels VNTR probe. Place the nitrocellulose filter paper in a solution of radioactive labeled single-stranded DNA probe. DNA probe binds to the complementary sequence of the DNA fragments on the membrane to form hybridized DNA. And finally, detection of hybridized DNA by autoradiography. Hybridized DNA is photographed on an X-ray film called autoradiography. The autoradiogram gives an image in the form of dark and light band. It is called DNA fingerprint. Then, DNA fingerprint differs in everyone except in monozygotic twins, that are identical twins. The sensitivity of the technique can be increased by use of polymerase chain reaction PCR. Therefore, DNA from a single cell is enough for DNA fingerprinting. Then at a glance, first of all, isolation of DNA, digestion of DNA by restriction endonuclease. As you can see the steps. Third one is the separation of DNA fragment by gel electrophoresis, long DNA fragment, short DNA fragment by gel electrophoresis. Fourth, transferring DNA fragment to synthetic membranes, nylon. Fifth, hybridization of using radioactive labeled VNTR probe like this. And sixth one is the detection of hybridized DNA by autoradiography. Applications: forensic tool to solve paternity, rape, murder, etc. For the diagnosis of genetic diseases, determine phylogenetic status of animals, and the fourth one is the determine population and genetic diversities. And in forensic tool to solve same points are given. Here you can see how it is changed. Like it is a mother one, child, father is claiming, three are there, and as you can see mother and child. Here father 1 and person 1 is likely to be the father of the child. So with this DNA fingerprinting work. Then again, solving crime with the help of DNA fingerprinting. This diagram I think is from NCERT. Let's see your NCERT what it says here. DNA fingerprinting involves identifying differences in some specific regions in DNA sequence called repetitive DNA. The bulk DNA forms a major peak and other small peak are referred to as satellite DNA. And there are two satellites, micro and mini satellites. Polymorphism, and here variation at genetic level arises due to mutations. Then here it is given, if an inheritable mutation is observed in a population at a high frequency, it is referred as DNA polymorphism. And fingerprinting was initially developed by Alec Jeffrey. Use the satellite DNA as a probe that shows the very high degree of polymorphism. It was called VNTR, variable number of tandem repeats. First of all, isolation of DNA, digestion, separation, transforming, hybridization, detection of hybridized DNA. The size of VNTR varies in size from 0.1 to 20 kb. It differs from individual to individual in a population except in the case of monozygotic or identical twins. And PCR is given, polymerase chain reaction. We will study about this in chapter 9, and the diagram is there as you can see. And with this chapter is completed. Some about this topic and particular DNA fingerprinting, we will go through this in the chapter 9, where we can easily identify what is PCR, how it works. So thank you all for watching this video, and new viewers, please subscribe my channel for latest videos of mine, and do not forget to like, comment, and if you find any doubt, do contact me. My number is mentioned in the video. Thank you. Jai Jagannath.